Assalamualaikum warahmatullahi wabarakatuh. My name is Intan Nur Hannani Binti Abdul Halim and I am from AS20168. Today, I'm going to present about my industrial training at Crown Research Sdn Bhd. Next, I'm going to talk about the profile of the company. Crown Research Sdn Bhd is located at Lot G147 Block 14, Jalan Sultan Tengah, Putrajaya, 93050 Kuching, Sarawak. And the phone office is 082-446 and 489, and the operating hours is from 8 a.m. to 5 p.m. from Monday to Friday. Crown Research Sdn Bhd is a research and product development company that consists of 78 staff. This company categorized as government-linked company (GLC) that have a primary commercial objective and in which the Malaysian government has a direct controlling stake. Crown Research Sdn Bhd is a research and product development company that consists of 78 staff. This company, categorized as government-linked company, GLC, that have a primary commercial objective and in which the Malaysian government has a direct controlling stake. And this is the organisation chart. It consists of the board directors, the chairman, Yang Berbahagia Encik Kamri bin Haji Effendi, the director, Yang Berbahagia Puan Dayang Erni binti Awang Hashim, and director Yang Berbahagia Encik Yusuf bin Sobeng, and followed by the management team, the CEO Haji Zaidil Haji Husaini, followed by Dr. Bala Ana Jamil, Zahri Peri, Azri Muhammad Razali, and Dr. Nalaina Yunus. The activity of the company can be divided into 4 parts. First is the upstream technology, where they focus on breeding and propagation, soil and water management, agronomy, crop protection, and mechanization and post-harvesting handling. Next is the downstream technology, which they focus about analytical studies, mining technology, byproduct utilizations, and product improvement and development. Next is the techno-economy, which identify and explore the potential business and commercialize sago products and services, monitor and evaluate the developments of entrepreneur carrying Crown's product, transferring technology of sago upstream and downstream, research for commercialization, and conduct the promotions for marketing of the developed products from sago research. And lastly is the corporate services, which they focus on administration, finance, human resource, and information resource. These are the examples of organization manufactured, where they have MD2 pineapple and MD2 pineapple crown. Next, they also manufactured Carnera sago cookies and Carnera sago pearl. The objective of the organization is to be at the forefront on crops and product research and innovation focusing on sago. Next, advance in sago industry through effective research and innovation. The industrial training objective first is to experience first-hand working as a researcher in an actual working culture and industrial practice. Next, to apply technical skills, knowledge, and engineering method to a real-life situation. Then, I will be able to work alongside other professional researchers of different areas of expertise. Next, to develop responsibility, self-confidence, spirit of teamwork, communication skills, both oral and written, and industrial approach of solving problems. Next is to witness the functioning and organization of business and companies, to expose to safety practices and regulation in laboratory, and lastly for preparation to converge the workforce upon completion of studies in University Technology Mara. Next, I'm going to describe the industrial training objective to meet organization objective, in which I will be able to learn on how to isolate DNA of sago palm as the organization is a research and product development. These are the works that I have done during my internship. First is DNA isolation of sago palm, and in the picture you can see me taking liquid nitrogen to be poured in a porta and metal that contain sago palm. Next is DNA isolation of pineapple, and in the picture I am preparing fresh buffer solution for DNA isolation. And third is the gel electrophoresis, and in the picture it is the DNA that have been separated. And lastly, subculturing sago palm, and in the picture there are the plants that have been subcultured. Next is a planting out plant, and in the picture I am transferring plant from a test tube into a nursery pot. And next is preparation of lysogeny broth, LB agar, and lysogeny broth agar and ampicillin. And in the picture, I am streaking a plate with inoculating loop. And lastly is a media preparation, and this is where I prepared for subcultured media. These are the works that need to be performed the remaining period of industrial training from week 12 to week 14. First is DNA isolation of 60 samples of sago palm, where I will be separating DNA by using gel electrophoresis and replicating DNA by using PCR machine. I will be also planting out sago palm in the nursery. This is the value added that was obtained. First is boost self-confidence. I am now able to communicate with others and not afraid to ask for help. I can also finish my task by myself because I am now confident with my work. Next is stronger social and professional network. Industrial training provides me an opportunity to learn from the professional in the industry. Thus, this will increase my social skill. Next is to explore a career path. This particular training allows me to work in the desired field, helping me to decide if the field is right for me. I am now more likely to feel confident after done with the internship. And lastly, to gain valuable work experience. The hands-on work experience I had received was invaluable and cannot be obtained in a classroom industry, making this one of the most important value that I have obtained in the internship. The value added that had been contributed to the organization first is to submit high quality of work. I am able to perform subculturing of sago palm without contamination. Next is to bring new perspective and fresh ideas into the work environment. By providing positive reinforcement, the others will make them feel encouraged to perform in work. And lastly, to supply quality of support and energy. To provide consistent support to the officer by doing the task before the deadline of the submission. This is the new processes or procedures that were obtained from the organization. First, I am able to perform DNA isolation of sago palm and pineapple. Next, I am able to analyze DNA by using UV spectrometer. And this is the new processes or procedures were contributed to the organization. I am able to finish the project of DNA extraction of sago palm and pineapple on time. The relationship achievement in industrial trainings organization is gain more trust and respect among us. Now I get the trust given by my supervisor to do a task by myself. My suggestion for improvement of industrial training. First is the company should provide any form of reward on the work done by the intern. Next, to change the attendance of using logbook into fingerprint attendance system. Because attendance system is more accurate for the intern to clock in and out, it is also good to maintain a high level of security and against the theft of sensitive material. Next, I would like to recommend the company to upgrade the lab equipment for better laboratory integration. In conclusion, the training able to expose the intern to the real-life working environment before graduating from UiTM. Next, the intern get a lot of skills and learn something very useful and relevant to biology courses. Moreover, it allows the intern to adapt to the association between colleagues. And lastly, it trains the intern how to communicate effectively. For acknowledgement, I would like to express my gratitude to the Industrial Training Supervisor, Dr. Bala Ana Jamel, and other Crown Research Upstream Technology Division, Puan Evra Rauni binti Ibrahim, Puan Syamsah binti Hamid, Puan Lenis binti Iman, Puan Hasnah binti Satem, Puan Herma binti Aman, and Encik Muhammad Tirmizi Abdul Ghani. And that is all from me. Thank you.